Go.
Hello, my name is Garrett Moulton, and this is my lab 6 for mitosis and meiosis. As you can see, this is my work area. I have my list on the computer screen. Other than that, just a drawing for mitosis and my pipe cleaners. So, for mitosis, there are 6 different phases. In the interphase, there is the first gap where all it does is start to gather the building blocks, all the different proteins, and the energy it needs to use mitosis. Then there is the S phase where it creates sister chromatids that look like these. It takes 2 chromosomes paired together. And then there is the G2 phase, which is the second gap that replenishes energy and synthesizes proteins for movements. After interphase, we move to the prophase, which is where the sister chromatids and all the other chromosomes start to condense and get more visible under a microscope and move towards the center of the cell. Then there is the metaphase, which is where the piece of paper right here is going to be my cell. They get in a straight line on an equator, is where they're going to start to try and separate. Then there is the anaphase, where they separate at the center and start to pull apart to the opposite poles of the cell. And during the telophase, they are at the opposite ends of the cell and they start to condense on the opposite sides of the cell and start to split across. The cell then starts to divide into 2. And once it divides into 2, that's the cytokinesis phase, where it's essentially 2 complete cells after that. And each phase has a certain amount of chromosomes in it. The interphase has 46, the prophase has 46, the metaphase has 46, anaphase has 92, the telophase has 92, and then once you hit cytokinesis, they split apart and they each have 46. Now, for meiosis, they go through essentially the same process. However, in prophase 1, with the sister chromatids, they start to cross over. When they cross over, you have the sister chromatids like this, and they cross over in one little section down here. When they cross over, each little piece breaks off and they tie into the opposite sister chromatid, causing 2 with opposite colors in one tiny section. And then they are the same as mitosis, metaphase 1. They line up on the equator of the cell. Anaphase, they start to split apart and go to the poles of the single cell as it starts to pull itself out. And then they hit telophase, which is where they're at the opposite poles of the cell, and they start forming back together to create 2 separate cells. And then prophase 2 is taking each cell, each of the 2 cells, and separating them again from the 2 to essentially create 4 of the same exact cells. So it goes through the same prophase, metaphase, anaphase, and telophase. So now you have 4 cells of equal amounts of chromosomes. And the chromosomes for each phase of meiosis, for prophase 1, you have 46, metaphase 1, 46, anaphase 1, 46, and telophase 1, 46. For prophase 2, which is after you split them, each cell starts with 23 chromosomes. And then prophase 2 and metaphase 2 each have 23 chromosomes. And then anaphase 2 and telophase, during anaphase 2, it doubles to 46 and keeps 46 all the way through telophase 2. And after telophase 2, they split again, and you have 4 cells with 23 chromosomes in it. And that is it for my mitosis and meiosis video.